So as we start our notes for 14.2 on transcription and translation, take a minute and write one or two things under what you already know, and one or two things under questions you already have, or questions that you might have. Another minute or so on that, please. About 30 seconds. Hopefully everybody's taking a minute or two to write down questions and things that they might already know about protein synthesis. All right, ladies and gentlemen. So hopefully you had a chance to write down one or two things that you know, questions you might have in the K and the W part of our KWL. Now, ladies and gentlemen, throughout the presentation, there are going to be a handful of short videos. During that time, as we are watching the videos, I would like for you to write one or two things in the things that you learned area. So after every video, we are going to come back to this KWL section and we are going to write down one or two things that you learned from the video. All right, so we're going to go ahead and get started with our notes, ladies and gentlemen. And again, we're looking at protein synthesis. Again, the entire point of protein synthesis is this idea that we are building proteins. And we are building proteins using instructions from our DNA, using our RNA as the little worker bees, if you will, because proteins are super important to every living thing. All right. So we already did the K and the W. This is just review. This is not part of your notes, by the way. These are some things that you maybe wrote down in the stuff that you know about protein synthesis. Maybe you wrote some stuff down about DNA. Please remember the key things about DNA is that it is found in all living cells. And our three main things that we keep talking about when it comes to DNA is that it has three essential functions. The three essential functions of DNA are that they can store genetic information, they copy genetic information, and they transmit genetic information. Now, this is review stuff, guys. So if you didn't have anything in your K column, this would be great information for the things that you should know, because this is everything on this slide right here is review. The other thing that I want to talk about really quickly, because we know this, is the double helix base pairing. So we remember Chargaff's rule. In DNA, adenine and thymine always match. Cytosine and guanine always match. So we know that DNA has a sequence of nucleotides, bases, or codes. Somebody tell me, what do these bases code for? You can respond in the chat. You can respond.
Genetic codes.
Yeah, so what does our genetic code actually, what is it for? What is it coding for? What are we trying to make here, people? what does our DNA actually, what are the base codes, what are they coding for? If I have a gene, what is it coding for?
Um, DNA and RNA. Think.
DNA helps to make RNA, but what is the purpose? What is it? What is the actual thing coding for? Thank you, Matthew. I see you in the chat. Yes, it is actually going to be coding for a protein. Oops, there we go. Absolutely. The whole point of this is to code for proteins because proteins are the things that bring your traits into being. The fact that you have brown eyes, or maybe you have blue eyes or hazel eyes, not that I know because hardly anybody ever turns on their camera, but the color of your eyes is created by a protein. Some of you probably love cilantro. That is that green sort of leafy looking thing inside salsas like pico de gallo. Some of you guys probably think cilantro is disgusting and sort of tastes like soapy dirt. Whether you love cilantro or whether you think cilantro tastes like soapy dirt is because there is a protein that your body makes. Some of you might be lactose intolerant, meaning that if you eat dairy, bad things happen. Some of you guys eat dairy with no problem. The difference between being lactose intolerant, which is a trait, and the difference between being able to consume dairy, which is a trait, is a protein. All right, second question. What is a special molecule that puts all of this code together? What is the molecule that helps build this protein? Ooh, got very specific, Matthew. Amino acids. Okay, we're getting there. Getting there. I'm going to, who's the workhorse here? We have DNA. We have coding a protein. Who's actually
RNA. RNA.
There you go. So RNA is the one bringing this all together, ladies and gentlemen. All right, so we're going to watch a quick video. Again, I want you to go back to the learning part of your KWL in your note catcher, and I want you to write down one or two sentences, something new that you're hearing in this video. Okay. That is crazy. Sorry. There we go.
Grammarly helps you work more efficiently, which makes accomplishing your work goals easier than ever before. Get full sentence rewrite suggestions so you can say what you mean faster. Powered.
Stated Clearly presents What is DNA and how does it work? DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important, they're often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The neat thing about them is they can be attached to each other kind of like Legos to produce an endless variety of larger particles known as proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. Cells make up tissues. Tissues make up organs. And organs, when they're all put together and functioning, of course, combine to form living creatures like you and me. These proteins that make up our bodies and Keep in mind, there's millions of different kinds of proteins. They each have to be formed in the perfect shape in order to function. If they're the wrong shape, they usually won't work. That's where DNA comes in. DNA does a lot of interesting things, some of which we don't fully understand, but one of its main and most well understood functions is to tell amino acids how to line up and form themselves into the perfect protein shapes. In theory, if the right proteins are built at the right time and in the right place, everything else from cells to organs to entire creatures will come out just fine. This here is a simplified model of DNA. It shows us that the steps of the ladder are made up of 4 different kinds of chemicals shown here by different colors and letters. If you look at just one half of the molecule, you can read its chemical sequence or genetic code from top to bottom, sort of like a book. A single strand of DNA is extremely long, millions of letters long. It spends most of its life coiled up like a noodle, living inside the nucleus or the centerpiece of a cell. Amino acids, however, live outside the nucleus in what's called the cytoplasm. To help DNA interact with the cytoplasm and convert those amino acids into proteins, special chemicals inside the nucleus make partial copies of the DNA code. These partial copies, called RNA, look a lot like DNA, but they're shorter, of course, and they're missing one of their sides. Their small shape and size allows them to fit through tiny pores in the nucleus, out to the cytoplasm, and into the mouth of another particle called a ribosome. Ribosomes are protein-building machines. They read the RNA code 3 letters at a time, suck amino acids out of their surroundings, and stick them together in a chain according to the RNA code. As the chain grows, it bends, it folds, and it sticks to itself to form a perfectly shaped protein. Every 3 letters of the RNA code tell the ribosome which of the 20 different kinds of amino acids should be added next. For example, CAA tells the ribosome to grab a glutamine, AGU tells it to grab a serine, and so on. Once a protein is built, it can then go on to do a number of different things, one of which could be to help form a brand new cell. So to answer the original question, what is DNA? DNA is a molecular blueprint for a living thing. How does it work? DNA creates RNA, RNA creates protein, proteins go on to form life. This entire process, as complicated, as sophisticated, as magical as it might seem, is entirely based in chemistry. It can be studied, it can be understood. I'm John Perry, and that's DNA stated clearly.
All right, so take a moment or two and make sure you write down in your note catcher 1 or 2 things in the learned area that you learned from that video. I'll give you a minute. All right, so hopefully you had a chance to write some information down, something that you learned that was new. Would anybody like to share what they have learned? Or something new that they heard in the video that they hadn't heard before? Anybody? Anybody at all? I need one person to tell me something that they have learned in that video, and then we can move on.
I learned proteins and RNA and amino acids. Yeah, a couple of things.
Okay, cool. So again, And the central dogma of biology, because again, biology is the study of life, is this idea that we go from DNA to RNA to proteins. Without the proteins, we would have a really hard time not having life here on the planet Earth. All right. So this is going to be minus the chart. You do not have the chart in your notes, ladies and gentlemen. These are the first 3 bullet points, okay, on the first page of your note catcher. So before we can get into transcription, we're going to take a minute and we're going to go back and we're going to review a little bit about DNA, a little bit about RNA. Again, DNA is the architect. DNA is the recipe book. DNA is the blueprint. DNA contains the instructions or the master plan on how to actually build those proteins. Okay? Now, the RNA is the doer, okay? If DNA is the architect, then RNA is the builder. If DNA is the recipe book, then RNA is the chef. If DNA is the blueprint, then the RNA is going to be like the people at GM on the line that actually build the car, okay? RNA is the doer, DNA is the plan. RNA is the one actually out there producing, making, synthesizing the proteins. Now, another key difference here is that DNA always stays in the nucleus. DNA, if it's damaged, we know can lead to mutations, and mutations can lead to really bad health outcomes in most cases. So DNA, we try really hard as a living thing to keep our DNA safe. Part of keeping our DNA safe means it's going to have to stay in the nucleus. However, RNA is just a copy. We don't have to be as careful with copies. RNA then will travel from the nucleus out into the cytoplasm. If you have ever traveled to Washington, D.C., one of the cool things that you can see at Washington, D.C. is the original Declaration of Independence. And it is I don't remember where it was exactly. I want to say it was in one of the Smithsonian's, but it's behind this, like it's in this case and it's behind all this glass and it's super duper protected. And like they have moisture sensors and all this kind of good stuff on it. Because it's the original. They have to protect it, much like we have to protect our DNA. Now, I'm guessing like every copy of history book you've ever had has a copy of the Declaration of Independence in it. There's tons of copies. We don't have to be as careful with copies. So again, DNA is the original blueprint. It has to stay safe. It stays inside the nucleus. RNA, the copy, we don't have to be quite as careful with it, and it can travel out from the nucleus into the cytoplasm in order to make these proteins happen. Underneath here, we see that there are some things that are exclusive to DNA, things that are exclusive to RNA, but the similarities between them as well. I do want to point out that DNA and RNA are both nucleic acids, which means that they do have some things in common. But they also are unique. So they're going to have some unique things as well. I'm not going to belabor the point of the chart. We have done similarities. We've done compare and contrast on DNA and RNA in at least 4 different assignments already. So I just put another one here. Feel free to study it. And of course, this will be on the video, so you'll be able to go back and visit it as many times as you would like. Issues or questions? All right, hearing none, we move on. Okay, ladies and gentlemen, this is going to take you through the protein section of your notes, as well as the amino acid section of your notes. I will give you about a minute to write it down, and then I'm going to go ahead and talk about it because there's a little bit more stuff to write than before. So take a moment, get this stuff typed into your notes, and then we'll talk. so it's been a minute, so let's talk. All right, so again, this is just a little bit of review. We've talked about this before, but it is the foundational piece to understanding transcription and translation. So it's worth kind of a do-over, if you will. All right, so proteins are polymers. What that means is they're really, really, really big molecules. And we make these polymers, we make these protein polymers by joining together a bunch of amino acids. Amino acids are the building blocks of your proteins. Amino acids are held together by peptide bonds. So remember, we talked about covalent bonds, we talked about hydrogen bonds, we talked about ionic bonds, we've talked about polar bonds. This is just another type of chemical bond. It is called a peptide bond. Peptide bonds hold together these long amino acid chains. So we call them polypeptide chains because poly means many and peptide is the name of the bonds. We have many of these bonds holding these guys together. Again, why do we care about protein? They control our reaction rates. They regulate our cell processes. They build cell structures. They transport materials. Chances are, maybe you have somebody in your family. Maybe you know somebody. Maybe you've gone to school with somebody. As an African American community, one of the genetic diseases that affect African Americans far more disproportionately than other sections of the population is sickle cell anemia. What sickle cell anemia is, I mean, when you get down to the like the chemical part of it, is you have a malformed red blood cell. So people who suffer from sickle cell anemia have a really hard time transporting oxygen throughout the body because the red blood cells have the wrong shape. And the reason they have the wrong shape is because those individuals with sickle cell create proteins that are essentially mutated. So again, that transporting of materials, that's huge. And there are whole diseases, and like I said, in the African American community, the most prevalent one compared to other populations is sickle cell. But again, that's a protein problem because your body makes the wrong protein. It's not the wrong protein, it's just a malformed protein. And then, of course, proteins also help fight disease. Your entire immune system, everything from your T cells to your white blood cells, your entire ability to fight disease, whether it is the common cold, the regular flu, COVID-19, cancer, any type of disease, again, is based in proteins. Again, super important for life. We talked about amino acids previously. Amino acids contain an amino group, which we see pictured down here. Carboxyl group, which is the yellow dot. The amino group, which is the one with the N over here, and then the variable R group. That variable R group is what makes each amino acid separate, different. And we have 20 different variable groups, and that's why we have 20 different amino acids. Issues or questions, ladies and gentlemen? Hearing none. This is going to take us through our next section of notes, ladies and gentlemen, through RNA, through messenger ribosomal and transfer. Take a minute, write it down, type it in, and then we'll talk in a sec. Okay, so we know that RNA is like the working man, right? So, like, again, it is the doer. RNA is the working copy. It is the working copy of a gene segment of DNA. Remember, a gene is just a small section of DNA that codes for a specific protein. And then that RNA, because it's made that copy of that gene, is now has the instructions to make that particular protein. So that protein might be the protein that you need to make the color of your eyes. It might be to make lactase, which helps you digest dairy. It doesn't matter what it is. Whatever trait it is that you have, this protein that you're trying to make is going to help you manifest that particular trait. RNA synthesizes proteins. The word synthesize is just a fancy schmancy word for build. I don't know why biologists feel the need to use fancy schmancy words instead of like regular old words, but they do. So RNA is the one that actually brings protein synthesis to life. And again, it is doing this by controlling how our amino acids are put together into proteins. Now, there are 3 RNAs that need to work together in order to make a protein. And we know this, okay? We have mRNA, which is our messenger RNA. We have rRNA, which is our ribosomal RNA. And we have tRNA, which is our transfer RNA. These 3 forms of RNA work together to synthesize proteins. These these 3 types of RNA work together to build and assemble the proteins. Any missing part of this puzzle, proteins don't get built. Questions or issues? All right, hearing none. This is 3 types of RNA, ladies and gentlemen. They are going to perform different tasks, and we just said that, okay? And they are each unique, and they have their own special structure. Now, I don't know if I included this diagram in your notes or not. In fact, I don't think that I did, but that's okay. mRNA. How do I identify mRNA in a diagram? mRNA always has like a ribbon shape. It is the classic RNA shape. It is the single strand of RNA that we all see in all of our textbooks. Anytime we think of RNA, mRNA is the one we're thinking of. It's a nice, beautiful single strand half coil thing. Okay, that is mRNA. That is like the poster child. That is the poster model for RNA. Our rRNA is always kind of blobby. I mean, for lack of a better word, it's just sort of this blob because it is the ribosome, and the ribosome kind of has a blobby shape and whatever. No better way to put it. Sorry, it's a beautiful blob. Without them, we don't make proteins. Love the blob, but it kind of looks sort of blobby. And then the tRNA, how to identify it, it always kind of sort of has a little bit of a T shape. Now, it's not a perfect T, I'll give you that. But in the tRNA, we are always going to have an amino acid on one side. That's what this black dot represents. We have an anticodon on the opposite side. And an anticodon is sort of the complementary pair of a codon. And then, of course, in the middle, the blue is representing the actual strand of RNA. So it is like the mRNA, except for it's all folded up kind of funny and weird in this sort of T-like shape. All right. So again, they're each unique. They all have their own job, and they all have their own structure. So again, quizzes, tests, things like that, you will see similar pictures. But if you need to know the difference between the 3, RNA is always blobby. mRNA always looks like the classic RNA, and tRNA is always kind of folded up almost like a T. Sometimes it looks sort of like a fork. Sometimes it looks a bit like a plunger. These are common artist renditions of tRNA. All right, so let's take a quick look at mRNA, ladies and gentlemen. Give yourself a minute, copy it down, and then we will move on from there. All right, so again, this is a little bit of a review, but it's okay because we need to have a solid foundation of this in order to understand the transcription translation process. mRNA, messenger RNA, what it does is it makes a copy of a gene. Again, all a gene is a small section of your DNA that codes for a specific protein. For example, on your 15th, remember you have 46 strands of DNA in every cell in your body. You got 23 from mom, you got 23 from dad. We talked about that when we talked about cell division. On strand number 15, every single one of us humans has a small section of gene code that codes for our eye color. And it's just a tiny little section of it. If a DNA strand is millions of long, a single gene might be a couple of thousand. That sounds like a lot, but that's like nothing compared to millions. Anyway, so our mRNA is going to make a copy of a gene sequence from the DNA. That copy is what we call the transcription process. And we'll get a little bit more into that in a minute. mRNA, excuse me, is going to act like a messenger. It carries the instructions for making the protein. It copies them from the DNA in the nucleus. Remember, DNA never leaves the nucleus, and it carries it out into the cytoplasm in order for the ribosomes to actually build the proteins. It gets out of the nucleus through a nuclear pore. So the nuclear membrane has these little tiny holes in it called nuclear pores. And just like the pores in your skin, those little tiny holes that you have all over your skin that when you sweat, your sweat comes out of. Well, a nuclear pore has these little tiny holes and the RNA can come out of it. And again, it is going to carry the instructions from the DNA out to the ribosomes inside the cytoplasm. It is a messenger. Just like any messenger, it just carries the information. It doesn't do anything with the information. It just gets a copy of the information and carries it to where it needs to be. Issues or questions? Okay. This is the next section of your notes, rRNA, along with my really poorly created GIF down here, but that's okay. Take a minute, write it down, and then we'll talk. Okay, so proteins are assembled in the rRNA. The rRNA is what makes up the ribosome. For lack of a better explanation, a ribosome is made up of two subunits of rRNA, and it's kind of like a hamburger bun. Like you have a top and you have a bottom, and then like there's stuff in the middle. And the stuff in the middle is going to be like your mRNA and your tRNA, and we'll get into that in a minute when we actually talk about the translation process. But just understand that your ribosome is made up of two pieces of our RNA. And we can kind of see that in the diagram here. Subunit on top, subunit on bottom. And I've always described it kind of like a hamburger bun, like because we all know hamburger buns, right? You got the top, you got the bottom, and the stuff in between. And again, RNA is like the construction site. So we've all seen construction sites. Stuff is being built there. Same thing here. The RNA, the ribosomal RNA, this is the construction site for your protein. Issues or questions? Okie doke. And you can kind of see in the little GIF, it is building that protein. That's what happens in the rRNA. Okay. All right, ladies and gentlemen, tRNA. Take a minute, write it down. And this is the last slide that we are going to do today. Then I will check your attendance for anybody who came in late. And then we'll continue tomorrow. Nobody should be turning these in. We are not done with these notes. All right, so transfer RNA is going to transfer each amino acid to the ribosome. Transfer RNA is like the delivery system. It will literally go out, find the amino acid that we need to build the protein, and carry it back to the RNA, carry it back to the ribosome. And it's what it carries back is specified by the mRNA's code. All right, so again, here it is. We do not have our time for our video recap, which is okay. We can start there tomorrow. Excuse me. But just know that we have this kind of cross or T-shaped RNA with the amino acid on one side and the anticodon that's going to read the mRNA on the other side. All right, so I'm going to go ahead and stop our video.